You could train an AI on 30 million human cells to map which genes control which. Or you could measure how much each gene wiggles from cell to cell. One researcher ran both. The wiggle measure won. Single-cell AI models like Geneformer and scGPT are trained on tens of millions of human cells, inside a mechanism called attention assigned scores to gene pairs, weights that researchers have been reading as regulatory connections. High attention between two genes means one probably controls the other, or so the field assumed. Labs started building drug target pipelines on this, and the assumption was never rigorously tested. A 37 analysis, 153 test framework across 4 cell types now has an answer, and it comes from 3 tests, each one harder to argue with than the last. The first test is the most direct. Can AI attention scores predict which genes change when you knock out a specific gene with CRISPR? The study used a massive leukemia cell dataset, 640,000 cells with 2,024 genes knocked out one by one, each testing whether the AI could predict which other genes would respond. Figure 2 shows the head-to-head. Geneformer, 316 million parameters trained on 30 million cells, scored an OROC of 0.70 at predicting knockdown targets. Spearman correlation on a single dataset got 0.696. Statistically indistinguishable, but measuring how much each gene's expression varies from cell to cell, that single number scored 0.881. Variance alone, computable in seconds, beat the AI by a wide margin, and this held across all 27 parameter combinations tested. But variance being high might just be a proxy for what the AI already knows. The real question is whether the AI adds anything on top of what gene statistics capture. The incremental value test addresses this directly. Take the gene-level statistics, variance, mean expression, and dropout rate, and train a predictive model. Then plug in the AI's pairwise attention scores on top. If attention captures regulatory structure that simple statistics miss, prediction should improve. The dataset had over 500,000 gene perturbation pairs, enough to detect even tiny effects. Figure 3 shows the result. Gene-level features alone scored an OROC of 0.895. Gene features plus Geneformer attention also scored 0.895. The improvement was negative 0.004, with a confidence interval that just touches 0 from below. This is not a power problem. With more than 500,000 observations, the analysis had 99% power to detect a difference of 0.005. The pairwise network learned from 30 million cells contributes nothing beyond what simple gene statistics already capture. Maybe the scores matter in ways a regression cannot detect. There is a harder test, one that directly cuts the circuit. Delete the supposed regulatory heads entirely and see if the model notices. Geneformer has 324 attention heads across 18 layers. The author ranked them by how well their patterns matched known regulatory interactions, then masked heads out during the model's forward pass, measuring whether perturbation prediction changed. If the heads really encode regulation, cutting them should hurt. Figure 4 shows the dose-response curve. Cut the top 5 regulatory heads, nothing changes. Cut the top 10, 20, 50, that is 15% of every head in the model, still no degradation. Now ablate 20 randomly chosen heads. Performance drops from 0.704 down to 0.697, and the targeted heads were not being cut gently. They disturbed the model's output 23 times more than random heads did, so the surgery worked. The regulatory heads just do not matter causally. The signal is spread across the whole network, not concentrated in identifiable circuits. Now, the limits. Causal ablation only worked on Geneformer because of how it is built. scGPT could not be tested this way. T cells and iPSC neurons showed the same trend, but each had only 7 evaluable perturbations. Far too few for firm conclusions. The benchmark database, Trust, captures only a fraction of real biology. And ruling out attention does not rule out the full model. A transformer can be a powerful predictor without its attention patterns being interpretable readouts of causation. Where the actual regulatory signal lives remains open. There is a constructive result. A method called cell state stratified interpretability, CSSI, fixes a specific failure mode. When cells are too heterogeneous, pooled attention scores dilute and become noise. CSSI clusters cells into similar groups first, computes attention within each group, then combines the results. This chart shows it holding near its ceiling while pooled attention degrades, improving network recovery by up to 1.85 times. That is a real tool, but still no fix for the core problem. These models were supposed to have learned the language of gene regulation from tens of millions of cells. What they actually learned is which genes tend to be busy at the same time. That is genuinely useful, and genuinely not the same thing as knowing what controls what.